Good morning. Good morning, everyone. Happy Monday. I hope everybody's doing well. I hope you had a nice weekend. Chilly weekend for sure. That's a little better. All right. As usual, we are waiting for a few people to join us. Go ahead, if you will, and type your first name into chat so I can see that you're here. Thank you. Thank you very much. That's great. Thank you. That's my record of attendance for people. So I appreciate that. All right. So this morning we are going to be talking about a couple of things. Our lab exercise for today is about examining living organisms, specifically living microbes. So we'll be starting that today. We probably won't finish it. We'll finish it up on Wednesday when we get together. But before we get into that, the very first thing I want to do is ask if anybody has any questions about lecture material that you watched last week. If any of you have started looking at this week's lecture material, you can certainly ask about that as well. I did a little bit of grading this morning, so I could see that I got all, if not all, maybe all but one of the lecture quizzes submitted. I saw that everybody completed their lab homework questions, which is wonderful. That's what I really like to see. So remember, this week, your due dates change a little bit. This week now, you'll have your lecture quizzes due during the week, Wednesdays and Fridays, and you'll have your laboratory work, your lab questions due Sundays. And we'll then stick to that schedule for most weeks. I just wanted to remind you, because you're able to go in and out of your lab homework questions as often as you want before you submit them, I just want to remind you that when you are done, when you feel like your answers are good, you do actually have to submit them. You do actually have to click where it says, finish or whatever and submit. Most of you did that, but a couple of you didn't. So the message that comes in on my end is that it's pending, that your work is pending, but I can see that you have answered all the questions. So I think it's just a matter of you not going the full way to submit your work. So obviously with the quizzes, it's different. You only get that one chance. So as soon as you're done, you submit, but just remember that you do have to do a submission step with the lecture homework. I mean, with the lab homework when you complete it. All right, very good. Let's talk for just a minute about that lab homework. Actually, let's talk about the quiz first. I haven't really gone through everyone's lab homework, so I'll hold off on that. But let's talk about the quiz for just a minute, because there were a couple of questions that I saw show up more than once that people had a little bit of difficulty with. First things first, remember that when it comes to microbes, there are microbes present in all three of the domains of living things. So there are microbes in the bacteria domain, in the archaea domain, and in the eukarya domain. We tend to think of eukaryotes as being like us, as being multicellular organisms. And of course, there are many multicellular eukaryotes, but there are also single-celled microscopic eukaryotes. There are single-celled microscopic eukaryotic organisms. So that qualifies them as being microbes. Think, for example, about algae or certain types of fungus or protozoa. There are different life forms in the eukarya domain that meet our definition for microbe. Now, remember too that bacteria and archaea are prokaryotes. This is where the language gets a little tricky. Bacteria and archaea are classified as prokaryotic, and that tells us that they are single-celled and they have no organelles inside of them. And remember, all an organelle is a membrane-bound compartment inside a cell. So anything, any little compartment surrounded by membrane inside of a cell is an organelle. And prokaryotes don't have any of those. So if it has a nucleus, it has to be a eukaryote. Remember, the nucleus is where eukaryotes house their genetic material. And if you're a living cell and you're on planet Earth, your genetic material is DNA. So a prokaryotic organism doesn't have a nucleus. Their DNA just sort of floats in the cytoplasm. Okay, so that was one of the things I saw in the quiz. The other question that came up on more than one quiz was about the question about the cell wall material. That was a hard question. Different types of microbes have different material in their cell wall. Now, some microbes don't have a cell wall. So any type of animal cell, and remember, there are animals who are microbial. Any type of animal cell doesn't have a cell wall. Our cells don't have any wall material surrounding them, but bacteria have a cell wall. Archaea have that S layer, and other types of microbial organisms, eukaryotic types, have different materials in their cell wall. So bacteria, all bacteria, use that very unusual carbohydrate called peptidoglycan in their cell wall. Some of them have a big thick layer of it, some of them have a little thin layer of it, but they all use peptidoglycan. It's a carbohydrate, it's very unique, it doesn't appear anywhere else in nature, and that's what's in their cell wall. Archaea don't have a cell wall, technically speaking, but they do have this layer of material around their membrane, and that's called an S layer. We talked about S layers briefly in that lecture last week. Now, when it comes to an organism like a fungus, and remember, there are microbial forms of fungus, they use again, another very unique carbohydrate called chitin in their cell wall. Chitin is very strong. It's not as strong as peptidoglycan, but it's still pretty strong when you put it to the test in the laboratory. It's the same material that insects use in their exoskeleton. So it's the same material that, would make certain types of insects crunch if you stepped on them. So it's strong, and that's what we find in the fungal cell walls. Now, the one group that is really different is the algae. Remember, algae are protists. They fall into a group sort of all their own. They just don't fit neatly anywhere else. There are many different species of algae, and it depends on what species you are looking at as to what material is in their cell wall. Some algae have cell walls that look like plant cell walls. They have cellulose around their membrane. But other forms have other very unusual carbohydrate materials. Some algae have a cell wall made out of agar, the same agar that we use for growth media in the microbiology lab. Some of them use a carbohydrate called carrageenan, and there are other types of carbohydrates that you'll find as well, depending on the species that you look at. So many organisms share only one kind of material. So all bacteria use peptidoglycan, all fungi use chitin, all plants use cellulose in cell wall. But the algae are different, and the algae are different in lots of ways, which is why they're in their own strange group. Um, so you have to look at the species to know what cell wall material you're dealing with. All right, good. So I will continue to grade your first laboratory assignment this afternoon and perhaps into tomorrow, depending on how fast it goes and how much time I can take for it. But those will be graded soon, and you'll be able to see how you did on that first assignment. And remember, this week you'll have your next assignment based on this week's lab material. And again, lab homework questions will be due typically on Sunday at midnight. Now, this week is an unusual week lecture-wise, and that's because you have 2 lecture topics, but you have 3 lecture quizzes. Remember, we talked about that last week. So just make sure you know when those quizzes are due. Go onto Blackboard. I'm going to pull it up right here for you. See if I can get that to take. There we go. Go up onto Blackboard and into our course. You should be looking at our home page right now on Blackboard on your screen. I'm going to click on course content in the left-hand menu. Then I'm going to scroll down to week 2, which we're just beginning. And here's my to-do list. So I have 2 lecture quizzes due by Wednesday at midnight. I have 1 lecture quiz due Friday by midnight. And I have lab homework due Sunday by midnight. And remember, all your links are down here. I have not put up the file, the slides file, but I'm going to do that this morning for you, just in case you need them. Okay. All right, any questions so far before we start into our lab exercise for the day? Anybody have any questions? All right, good, very good. Looks like just about everybody's here with us now too, which is great. All right, so I'm going to start into our laboratory material now, which again is the examination of living organisms. You know, a lot of what we do in the microbiology laboratory, a lot of what we're examining are actually organisms that are dead. And we kill them because of the way we prepare them for examination. So for example, when I put some bacteria on a microscope slide and I stain it, which is a very common procedure, the procedure of placing them on the slide and doing what we call fixation, and then staining them with certain types of dye, that's going to kill those cells. And, you know, frankly, that's not a problem because we don't need them to be alive. We just need to examine them. There are times, though, when it makes sense to keep microbes alive while we're looking at them. And probably the biggest reason is because we're curious whether or not they are mobile organisms. We're curious whether or not they can move on their own power. So in today's exercise, in the first part, we're going to try to achieve several different objectives. Number one, we're going to talk about a particular function of our light microscope that is very good for looking at living microbes. And that's the function known as phase contrast. We're going to look at how a particular preparation called a wet mount is made. We're going to look at another technique for examining living organisms, and that's called a hanging drop technique. And we're going to talk a little bit about knowing the difference when you're looking under a microscope between a microbe that has real motility, a microbe that's able to swim under its own power, and a phenomenon that's called Brownian movement. So we'll talk little bit about that. And then in the second part of the exercise, we're going to together make and examine a wet mount, since the wet mount is the most common technique that we use to examine living microbial organisms. So again, we use a lot of stain in the microbiology lab, and we're going to practice, we're going to watch how different stains are used as we go throughout the semester. And the reason we go to the trouble of staining cells is because it lets us see them better. We can see them better whether we're using what's called bright field microscopy, dark field microscopy. And you'll talk more about those this week in your lecture material. It just makes them easier to see. It helps them stand out from other materials on the slide. And it also helps us see various structures in the cells. And again, as I said, the process of staining is going to kill the cells. It's going to fix them to the slide. In other words, it's going to make them stick to the slide. And the stains themselves are toxic. So the poor cells, they're just not going to survive that staining procedure. If we suspect that we might have a microbial organism, though, that is mobile, that is capable of moving under its own power, we can't go through staining to examine it because we're going to kill it. We won't know if it was able to move. Now, when do we suspect that we might have mobile organisms? Well, first of all, there are lots of mobile bacteria, and a lot of mobile bacteria live in our bodies. So, for example, there are fecal bacteria in our intestine and in our colon who have flagella, who have those structures that are almost like tails coming off of their cells, and they allow them to swim through their environment. So we have organisms in our body that are able to move, but there are also lots of organisms out in the natural world that are able to move. And part of the work that gets done in a microbiology lab is examining these kinds of environmental samples. So yes, we do examine biological samples. We look at patient samples all the time, but we also look at environmental samples. And in fact, there are laboratories that are devoted to environmental microbiology. For example, state laboratories like the one we have in Concord have entire laboratories devoted to environmental microbiology. It means that there are times when we want to know what kind of microbes are in our bodies of water, in our lakes, in our rivers. You know, here in New Hampshire in particular, we have lots of recreational bodies of water. People swim in them, they boat in them, they fish. So we have to monitor those bodies of water to make sure that we don't get a sudden, unexpected growth of pathogenic microbes. Water treatment plants have to monitor. Now, that may seem a little strange to you, since if it's got sewage in the water, of course there are microbes in it. But water treatment plants actually have to check to make sure that their steps of water purification are working. In other words, that the number of microbes is going down as the water passes from step to step to step. A lot of people in our state and around the country and around the world don't have access to municipal water. They don't have water pumped in or piped into their homes. They use water tanks or water cisterns outside of their home, and those have to be monitored to make sure, again, that pathogens aren't growing in them. And finally, even things like ventilation systems have to be monitored. you don't think of a ventilation system as being water, but they're very humid. They have a lot of condensation in them, and they have to be monitored to make sure they're clean and that the building isn't sending pathogenic microbes in the air into all of the different rooms in the building. So there's lots of environmental applications for examining microbes without staining them because we want to see if they're capable of swimming. Now, I said at the beginning of the of our time today that there's a property of a light microscope called phase contrast. Our light microscopes are capable of several different modes of operation. In other words, we can look under what's called bright field. That's the normal way you look under a light microscope, or I should say the most common way you look through a light microscope using bright field. But you can also turn a knob on your microscope and go into dark field, and you can go into what's called phase contrast. Now, phase contrast is the best mode to use when you're looking at living things, living microbial cells. And that's because we're not staining them, so they're going to be hard to see. If we were to just put them under the light field or the bright field mode, they'd be pretty hard to see because they would be the same color as the background. What phase contrast does is make them stand out a little bit more. Remember, living cells are three-dimensional. We tend to think of them as being two-dimensional, but they really are three-dimensional. And especially if they're mobile, we need a way to pick them out from the background. And one of the ways that we do that is by using phase contrast, because phase contrast makes cells have sort of a halo, a bright halo of light around them. As they move around, you'll see that they almost look like they're glowing. And that helps us distinguish what's a cell, a three-dimensional cell, from something else that might be on the slide. This is all because of this property that is used in microscopy called refraction. And again, you'll learn about refraction this week as you watch the lecture that includes the science of microscopy. All refraction is, in layman's terms, is the bending of light. And phase contrast is going to pick up small differences in how light bends when it interacts with three-dimensional living cells. It just makes it easier for us to see. And that's because you get these sort of bright rings around the cellular material. Now, on this slide, we're looking at one of those environmental samples that I mentioned a few minutes ago. This is a sample of water that came out of a recreational water body, a lake or a pond. And this is actually being viewed under the light microscope under what's called bright field. Again, the most common mode that people use microscopes in. The reason it gets its name is because the field or the background is very bright compared to the objects that we're examining. The objects are darker than the background. That's called bright field. Now, I want you to notice the incredible array of things that's on this slide. Lots of sizes, lots of shapes, and lots of colors. This is very typical for an environmental sample. You're going to get all different kinds of organisms out of a living body of water, like a lake or a pond or a river. Now, this particular sample was put through a centrifuge. So all of the solid materials would be gathered together at the bottom of the tube, and then they put those solid materials onto a microscope slide. And over here, what we're looking at is a 20x. We're looking through a 20x lens. So some things are very big, some things are very small, relatively speaking. Over here to the right, we've gone up to the 40x lens. the high dry objective lens, and you can see we see things a little more closely and a little more detail. I want you to notice these little black dots that you can see over here and over here. These black dots are actually bacterial cells. That's how small bacteria is under a 40x lens. We can't really see bacteria in detail at all until we go under the 100x objective lens. But you can see them under the 40x lens. You can see that they're there, these little dots. Now, all these other things then are not bacteria. These are eukaryotic organisms. Most of them are algae. There's some fungi in here, but just a collection of living things. And again, if you're doing laboratory work where you're examining samples to see what kind of microbes are in this lake or in somebody's well or in somebody's drinking water, this is the kind of work, these are the kind of samples you're going to be examining. Now, on this slide, what you're looking at is this preparation called a wet mount. Remember, there's two ways that we examine living microbes in the microbiology lab. We use a wet mount most commonly, and we can also use what's called a hanging drop technique. We're going to talk about both of those today. But I want you to look at this slide. First of all, you're going to notice that the background here is blue. And so you would be forgiven if you thought, oh, well, this slide has been stained with something. It actually has not been stained. This is a wet mount, so no stain. But there's a filter that is applied here just so it can be a little better visualized, because this is a short video clip. Now, I think you can make out that there are little spots on here, little dots all over this slide. If you look very carefully, these are bacteria. This is a type of bacteria that is mobile. So I'm going to go ahead and play this short video clip. And I want you to see first under 40x what this looks like. And then within about 30 seconds, we're going to see that the microscope goes up to a 100x lens, and we'll see things even more clearly. So right here, we're under a 40x objective lens. So we can see bacteria, but not easily. Now I want you to look carefully, and you're going to see motion on this slide. Not easy to tell much other than that. But under 100x, we can really see the motion more clearly. This is an organism called Enterobacter aerogenes. And in just a moment, we're going to see another one called Rhodobacter spheroides. And these are organisms that can be found both in our bodies and in the environment. But see them swim. These organisms have flagella. They have a tail-like structure that allows them to swim through their environment. This is the Rhodobacter. Don't worry about writing that name down. That's not important now. But understand that these cells are alive and kicking. These cells are swimming around. Not all of them, though, right? Some of them are not swimming. Some of them are likely dead. But a lot of them are just tooling around through their environment. This is what motility looks like under the microscope. A couple of features. Hold on, we'll go up to the 100x again in just a moment. The features of true motility are that all of these organisms are moving in different directions at different speeds. They are their own individuals, so they are doing their own motion. They're going their own direction. They're going their own speed. Some of them are slower. Some of them are faster. Some of them are changing direction. But it's really all over the place, and that's because they are individual organisms. That's going to be an important thing to understand about motility because, as I said at the beginning today, there is something called Brownian motion, which can trick us into thinking we're seeing motility when we're really not. Here's another one. This is a particular organism called Listeria. You may have heard of Listeria. The scientific name is Listeria monocytogenes. I've got the name down here for you just in case you're interested. You don't need to put that into your notes, but I want you to notice that this motility looks just a little bit different. Listeria has a weird way of moving. It's called tumbling motility. And if you focus in on one cell, if you put your attention on one cell and watch it, they tumble. It looks like they're doing somersaults. They still have the other features of mobile cells in that they're still moving at their own speed. They're all moving at different speeds in different directions. But notice that weird tumbling motion. That's unusual. And that's a characteristic feature for Listeria. Listeria is a pathogen. Listeria causes terrible food poisoning. If you've ever known anyone who's had listeriosis, it's not a pleasant disease to have. Listeria is primarily contracted through soft cheese. So things like brie and certain types of goat cheese, softer cheeses, and also other milk products. So if they haven't been correctly pasteurized and things like that, you can get listeria growing in them. So now I want to switch gears for just a minute and talk about how we make these slides. So let me ask first if anybody has any questions so far. Again, there are times when we're just going to go right ahead and kill cells and examine them because we don't need to know whether they're mobile. What we need to know is other features. Is it what's the shape of the organism? What's its gram status? What kind of metabolic capabilities does it have? But there are specific times when we suspect we have mobile organisms, and so we need them to be alive so we can look for them. We can look for them swimming. Okay. So again, two ways: wet mount and hanging drop. Wet mount is the more common method. It works really well for examining living specimens, and you can see motility. But hanging drop is the better technique for seeing motility. Let me say that again because that's important. Wet mounts are very common, the more common technique for examining living things. They're perfectly good for looking for motility. I have plus or minus here because they're not the best way at looking for motility. A hanging drop preparation, a hanging drop slide is a better method for looking for motility. But as you're going to see today, the hanging drop's a little bit harder to prepare, and so people tend to use the wet mount first. Now, a couple things to know about preparing samples using a wet mount or a hanging drop technique. You've got to mix the sample first. You really should mix all samples, but it's especially important if you're using a sample that's very dilute, like a water body, a water sample that was taken out of a lake or from the water treatment plant or from the faucet. It's very, very important that those samples be well mixed. What we're going to do after we mix up the sample is we're going to use a sterile instrument to remove some material out of our container. And we're going to choose the center of the container as the place where we'll draw from. We're not going to just put our loop into the very top. We're not going to put it all the way down to the bottom. We're going to put it right in the center. That's the best place to sample. And we're going to put our drop of sample right in the middle of our glass microscope slide. It's good to imagine that you can divide the slide into 3 areas, and you're going to use the middle third for your sample. That's a good technique for where to put your sample, how much to spread out your sample. You want to use about the middle third of your slide. And the other thing I tell students is imagine there's a quarter a coin, a quarter in the center of your slide. That's about how much you want to spread your sample out to the size approximately of a quarter. Remember, anytime you're making slides in the laboratory, you want to label them, especially if there's a possibility that you're going to be handing that slide off to someone else. It's important that all those things are on the slide that we mentioned. What are your initials? What day is it? What is the sample? What's the sample that's on the slide? So we have instruments in the lab to mix up our samples. This is a little vortex machine. These are very common in chemistry labs and in biology labs. You simply take your sample capped, of course, and you put it into this little cup and press down, and the machine will essentially mix. It will vibrate that liquid in order to mix it up really well for you. You can use an inoculating loop to take material out of a sample, but more often we use these. This little device in the center of your slide is called a transfer pipette. These are plastic single-use items. They come sterile or non-sterile. And if we were taking out a sample in the micro lab, we would want to use a sterile transfer pipette because we wouldn't want to accidentally introduce contaminating microbes into our tube. So you pull up a small amount of fluid and you pull right from the center. You can see this is a large sample of fluid, maybe 100 mLs are in this beaker. After it's been well mixed, you want to put your device in, whatever it is you're using to withdraw the liquid, and put the tip right around the center level of the fluid to get your sample. You want to put a drop of your sample onto your slide. Now, when students are first learning how to make a wet mount, they often put too much. It's very common to put too much, 2 or 3 drops. And what happens is you've just got too much fluid on your slide. It's not the end of the world, though. If you ever get too much on there, you can always withdraw a little of it by using a Kim wipe. Remember those green boxes that have Kim wipes in them? The Kim wipe will draw up, it'll suck up the extra fluid through capillary action, and you'll get down to a small enough volume. The reason you want a small volume is because you only want that fluid to be underneath your cover slip. And that's what you're looking at here in this drawing on the right. This is the microscope slide, and the artist has drawn it so that we're viewing it from the side. So you can see the droplet right here of the sample. And here's that thin piece of glass that we call a cover slip. We're going to place it on top of the drop, but we're not just going to drop our cover slip because you tend to get a lot of air bubbles that way. Instead, we're going to put one end of the cover slip on the slide, and then we're slowly going to lower the other end down, and that's going to help us get a nice wet mount. Now, let's talk for just a minute about limitations of wet mounts. The biggest one is that you only have about 15 minutes to examine that slide. You have a very limited time to examine a wet mount. And that's because there's a very small volume of fluid under your cover slip, and it's going to start evaporating immediately, especially after you put it on your microscope, under the light. The light is hot on a microscope, relatively speaking, not to you and I, but if you're a little organism in that fluid, it's going to feel hot. It's going to evaporate away that fluid pretty quickly. And within about 15 minutes, you're not going to have enough fluid left underneath your cover slip for a good examination. So if you're going to make a wet mount, you have to be ready to examine it immediately and quickly within about 15 minutes. Now, there is a little trick that some people use that will extend the life of your wet mount, and that's to put just a little bit of petroleum jelly, a little bit of Vaseline around the edge of the cover slip before you put it on. So you use a toothpick or something like that, and you just dab a little bit of petroleum jelly around the edges, and then flip it over and put it on top of your drop. That's going to help keep the air out. It's not going to make it last forever, but it might last half a day for you. So people do this when they want to make their sample last a little bit longer. The other thing to remember though is if your purpose in making this wet mount is because you want to watch microbes swimming, you got to be careful not to leave it under the hot light of the microscope for too long, because even with the petroleum jelly there to save the fluid, that hot lamp, that light under the microscope is going to eventually kill those microbes. If they get too hot, they're going to die under that hot light. So it's best to examine wet mounts quickly. Now, what about these hanging drops? Like the name suggests, this is a technique where instead of putting the drop on the slide and then squishing the drop with a cover slip, we're going to allow the drop to hang down from the cover slip and not touch the slide. We're going to allow the drop to stay as a droplet, just like droplets hang off of branches and sticks out in the natural world. We're going to allow this droplet to stay as a droplet and hang down from the cover slip. And the reason we make hanging drops is purely because we want to see motility. So motility is visible on a wet mount, but it's most clearly seen with a hanging drop. Now, you need a little bit of special equipment to do this one. You need a special type of microscope slide, and it's called a depression slide. And that's because it has a concave depression cut out of it. And I'll show you that in just a moment. Depression slides are also sometimes called well slides, W-E-L-L. And that's because that depression is also sometimes called a well. It's a little cavity on the slide. You're going to need a cover slip, of course, and this time you will need petroleum jelly. Now, not only is the hanging drop the best technique for viewing motility, but it's also a really nice way to see cellular arrangements in a natural sample. And by that, I mean some organisms like to associate with other like organisms in a certain way. They like to form certain geometric arrangements, and we can see that in a hanging drop. So there's a couple things to look at on this slide, and I know the writing is a little bit hard to see, but let's talk through this together. First of all, over on the right, this is one of those depression slides, and they're holding it in a way so you can see this circle right here. This slide started off with a flat surface and then essentially a concave depression was carved into the glass. So this area right here, if you were to put your thumb in it, it's actually cut in to the surface of that microscope slide. This is the depression or the well in the slide. Now these glass slides, unlike most glass microscope slides, these are kind of expensive because they've been etched like this. They've had some work done to them. So you're going to spend a few dollars for each one of these slides. And in the laboratory, we use them again and again. We don't throw them away after one use. We disinfect them, we wash and sterilize them and use them again and again. If you go to the top left here, again, the artist has drawn one of these slides with us viewing it from the side. And you can see it's got that depression cut into it. Okay. Now, what you're looking at here is the cover slip, again, from the side. And between the cover slip and the slide is that petroleum jelly. So the petroleum jelly is there to hold the cover slip up off the surface of the slide, just a little, but enough so that a drop of sample hanging down from the cover slip doesn't touch the glass slide at all. It hangs into that well and it doesn't touch it. So when we put this under a microscope, we're going to be able to examine what's in this drop without the drop being squished. Down here, this is just another artist's drawing of a hanging drop. And this one we're looking at from the top, just so we can see that the drop is in the center of the cover slip and the petroleum jelly is around the edge. Now, part of the reason people don't make hanging drops very often is because there is a little bit of art to making them, and I'll show you that in just a minute. But they are the best way to look for motility in a sample. What you're looking at in this slide on the left is a drawing of what it looks like to look at a hanging drop under a microscope. Now, this can be either under the 40x lens or the 100x lens. Because remember, we can see bacteria under the 40x, we just don't see them in much detail. What the person does who's looking through the microscope is they adjust the slide to the place where we're not looking at the drop entirely, we're looking at the edge of the drop. Okay, so in this drawing, this is the drop right here. Here's the edge of the drop. And then over here, this is just extra space on the cover slip. There's no sample over here. And this is what it actually looks like under the microscope. Here's the edge of the drop. This is going to take a different shape, of course, with every slide. This is the edge. So this is the liquid drop. We're looking at it at its edge. And then over here, this is just the rest of the cover slip that we're viewing. Now, this is a short video. It's about 2 minutes, and this one I didn't make, but this is a video that shows a person making hanging drop preparations. So this person is working under one of those biological hoods. I don't think they're working with anything pathogenic. I think it was just for ease of demonstration. Now, the first thing I want you to see is that they don't have gloves on. Horrible, horrible technique. You would never do this without gloves on. But again, I think this video was made just for teaching purposes, and maybe that's why they didn't. In other words, maybe there's no bacteria in here. But I still think even when you're just doing demonstrations, you should demonstrate correctly. So anyway, big problem here with no gloves. But I want you to see that the person has set out 1, 2, 3 or more glass slides that are depression slides. And next to each one, there's a cover slip. Now, on a couple of these, they've put some material right around the depression itself, right around that well. Don't worry about that so much. They were trying to demonstrate a couple of different things in this video. But what has to happen here to do a hanging drop is you have to put a little lip of petroleum jelly all the way around the edge of this cover slip. It doesn't go on the slide, it goes on the cover slip. You're going to take a drop out of this, a drop of your sample after it's been well mixed, and you're going to place it on the cover slip. Okay? Then, instead of moving the cover slip, which would be kind of hard because they're very little and they're very fragile, you'll pick up the slide. You'll turn the slide upside down and press it on top of the cover slip, and then flip the whole thing over, and you'll be able to examine that drop. So let's take a look at this short video again. Here you'll notice the operator is putting just a drop of liquid. Again, it's going to be much smaller than you think it needs to be. It's just a drop. They're using an inoculation loop to do that. Now we have to get a little bit of that Vaseline around the edge. Oh, and she's sterilizing her loop. Very good. Before she puts it down. Here's her little Vaseline container. They're calling this paraffin wax. That's fine. Now she's putting it on the slide. Again, don't worry about this. She's showing a couple of different techniques here. But what I want you to think about is putting it on the cover slip, because that's the correct way to do this. So she's going to put some wax or some goo of some kind on her cover slip. She's going to turn the slide over. She's going to flip it. Now she has a drop hanging. She's going to have a drop of material hanging from her cover slip down into the well. Press the slide on, flip it over. Listen, people who do these as their job, people who work in labs where they have to make a lot of hanging drops, they get really good at it. I am terrible at making hanging drops. I always manage to put either too much fluid on it or to get the drop to touch the edge of the glass microscope slide. So it's not easy. And of course, the next thing she's going to do is she's going to focus her microscope. Remember, you always focus on your low power lens first. You use those steps that we talked about in the video. You roll the stage all the way up. Then you look through the eyepieces and you slowly start to roll the stage back down until your image comes flashing into your sight. And then you use a combination of the coarse and the fine adjustment knobs to get your image into focus. Once you're in focus under the low power objective lens, you can use this black wheel here to turn the next lens into place, the 20x, and then the 40x, and finally the oil immersion lens with a little drop of oil on it. So she's going to take a look under her wet mount. She's going to get things focused. You can see her bringing the stage all the way up. Remember, we're going to look for, first of all, as the person using the scope, we're going to orient our hanging drop so that we're not just looking in the drop, we're looking for the edge of the drop. We're going to find the edge of the drop because all those living organisms are going to hang out right around here, right at the edge. So we'll be able to see in just a minute what hers looks like. She's coming into focus. Now, she's only under a low power lens here, so you can't see anything here other than the fact that she's right at the edge of her drop. You'll be able to tell where the drop ends and the rest of the cover slip begins because there'll be a color difference. Okay, and it's only going to be when she goes up to higher power, again 20x, 40x, 100x, that she's going to be able to see any motility. This is what we're looking for. We're looking for the edge of the drop. The living cells are going to tend to gather at the edge of that drop. It's just how it works when you have a drop in its natural shape hanging down from that cover slip. The living things are going to tend to gather at the edge, and that's where we're going to look for the motility. But of course, we'd have to go up to either 40x or 100x in order to see it. Now we are at 40x here, and all you can really see is a little bit of shimmering. It's just the video quality, but there is movement. These are all cells here coming up against this edge. So again, not real visible. The actual motility is not real visible in that video, but you can get a sense for how to make it and the fact that you want to look over at the edge. Now, true motility, you would think, is easy to see or not see, but there is a phenomenon when you look through a microscope called Brownian movement or Brownian motion. This is just a physical phenomenon that occurs when you're looking at something under a high magnification. What Brownian movement is the movement that happens in the natural world at that microscopic level, because molecules are constantly in motion and they are going to bump into each other purely through chance. Remember, every single molecule is always in motion. That seems strange to us, but until you get down to a temperature of absolute zero, even the molecules in your desk, even the molecules in your coffee cup are moving slightly. We can't see that motion, but it's happening. It's a vibration, in other words, that molecules have in the natural world until we get down to absolute zero. Now, when you're in a fluid sample, it's even more apparent. And what'll happen is things are just bouncing off each other a little bit because while we can only see the cells in the fluid, if we were looking under an electron microscope, we could see even smaller particles moving around, bumping into those cells, and making them bounce or shake or vibrate. This is not true motility. The cell is not moving under its own power. It's bouncing or shaking or vibrating because other things are bumping into it. It looks different from true motility, as you're going to see in just a minute. Brownian motion or Brownian movement is especially apparent under the 100x lens. So you have to know what it looks like so you don't get fooled into thinking that it's actual motility. The other thing to be aware of is that when you make a slide with fluid under the cover slip, that fluid is also going to move, at least at first, until it settles. So you're literally going to get fluid current moving under that glass cover slip. And you have to be careful because that fluid current is going to carry things along with it. And it's going to make it look like they're moving under their own power. Now, how do you know if you're looking at the fluid moving instead of the cells moving? When fluid moves, everything in that fluid is going to move along in the same direction at the same speed. That's very different from true motility, when everything will move in different directions at different speeds. Take a look at this short video clip here. We're looking underneath the microscope. Obviously, there's a bunch of strange looking things in here, and that's because this is a water sample out of a lake. What I want to show you here is the movement of the fluid current underneath the microscope. So I want you to look, and I want you to notice that things are moving in here. That's moving right there. These things are moving. This is moving. Does everybody see that? Let me do it again. Let me pull it back over so you can see it again. Things are moving, little tiny things, but they're moving. This is not motility. These things are being carried along by the fluid that they're in. How do I know? Well, because everything is traveling in the same direction at the same speed, approximately. All right. That's how I know that the water underneath my cover slip is just still swirling around. And if I wait a few moments, that will settle down a little bit. This, on this slide, is Brownian movement. I want you to look down here, right at about 4 or 5 o'clock here. These are a couple of yeast cells, again, in a water sample from a lake. And I want you to focus on them for just a minute and notice that I know the light is moving, but notice that these cells are shaking a little bit. They're vibrating. They're not still. There's another one up here that's also doing that. There's another one here that's also doing it. Let me show you again. I know it's hard because the light source is also moving, but these cells seem to bounce about a little bit. They're vibrating a little bit. That's Brownian movement. They are not moving by themselves. Things that we cannot see are colliding into them and causing them to move. Now, this final view shows both the movement of fluid current and Brownian movement happening at the same time. Here's some cells. They're vibrating and shaking a little bit, but they're also being carried along with fluid current. There's 1, 2, 3, 4 cells clustered together, and they are definitely moving across the slide, but They are all moving in exactly the same direction at exactly the same speed. This is not motility. If those four cells were truly modal, they would be swimming in different directions and they would be swimming at different speeds. They're also vibrating a little bit, if you can see it. It takes a little bit of practice, but remember, when you see a truly modal organism, especially under 100x, you're going to know it. You can tell that these cells are swimming. They're going their own way. They're not just vibrating in place. They're not just bouncing in place. They're going somewhere. They're moving across distance. And they're going in a way that's different from the way all the other cells are going. True motility has a very, I think, a very distinct look to it. All right, who has questions?
Okay, so
How long should you wait
Try to go one at a time if you can. Sorry.
They're celebrating there, even though you look like you could be.
How long should you wait to see if the slide still has current motion?
Okay,
Like how
so
long
it
should
sounds
you
like
wait?
I think we still have two people talking. So I hear a deeper voice, perhaps a male voice, although I'm not 100% sure. Why don't you go first, and then we'll let the other person speak. Who did we hear from just a minute ago? Oh, don't get quiet now. It's okay. I want to hear your questions. All right. Who was the female voice I heard?
That was me.
Okay, go ahead.
How long should we wait to avoid any fluid current or anything like that in the slide? Is there a way to, like a time limit?
So it's going to depend on how much fluid is underneath your cover slip, but you can certainly wait. It would be minutes. You can certainly wait, let's say 4 or 5 minutes, or you can go ahead and examine the slide and just ignore what the movement. Now you're going to have to sort of adjust the slide as things sort of flow across. But remember, they're not going anywhere. The farthest those things can go is to the edge of the cover slip. So a lot of people just don't even wait for fluid to settle on a wet mount. They simply examine it. And they use those controls underneath the stage to move the slide around and examine the cells that are on the slide. But if you find it distracting and you feel like things are being carried away and you can't find them, if you let it sit for 4 or 5 minutes, it will settle down. Good question. What other questions? All right, good. I tell you, when you're first learning in a microbiology lab, the movement of the fluid current movement and the Brownian movement can really make you very frustrated at the beginning. But once you get a chance to see 1 or 2 or 3 samples that have truly motile organisms in them, after that, it'll be much easier for you to distinguish between what's truly modal and what's not modal. In a lot of micro labs, you don't get to see mobile organisms all that much because most of the work they're doing is with stained slides. But depending on, if where a person works, especially if they work in a lab that looks at environmental samples, they're likely to see more mobile organisms. All right, good. We only have a few minutes left today. I'm going to pull our slide set back up here. What we're going to do the next time we get together on Wednesdays, we're going to pick up here. And what I've done is I've gone through the steps of how to make a wet mount. and I made a preparation for us that you're looking at in this picture here, in order to get some living cells here at home. Obviously, I don't have cultures growing at home, but what I did was I gathered a few strawberries from the market, and I mashed one of those strawberries up in a little bit of water, and I made a wet mount from that. Um, you might ask yourself, well, how did you know that there were microbes on that strawberry? And the answer is I know because strawberries are living things. And like all living things, strawberries have a microbiome. Anything that you buy in the grocery store in that produce section has microbes on it. Now, unfortunately, some of them can be pathogens, which is why it's always a good idea to wash your produce. But but even if you have a nice clean strawberry, it's still going to have microbes on it because the strawberry has a microbiome too. And one of the organisms that loves to grow on a strawberry is yeast. Now, again, I don't want you to be grossed out. If you're like me and you like to eat strawberries, those yeast are not going to harm you in any way, shape, or form. Those yeast are perfectly natural organisms on that strawberry, and they can become part of your intestinal microbiome as well. Perfectly natural, perfectly normal, healthy microbes to have in your gut. So there's no need to fear your produce. But yeah, it's a good thing to know, to understand that all living things have a microbiome. We all live in and among microbes, including plants. So they've got their own microbiome as well. So we'll pick up next time and we'll look at the yeast that grow on strawberries underneath the microscope in a wet mount. And then before that time, before we get together again, you can get started on your lecture material for the week. All right, if there's no other questions, I'm going to let you go. Have a good rest of your Monday, good start to your week, and we'll see you next time, okay? All right.
Thank you.
Have a good day.
Thank you.